Hello students, today I am going to discuss some questions related to your CBSE exam in the chapter Animal Cell Culture. So first question is like, why we use inverted microscope for animal cell culture? Generally we use inverted microscope for animal cell culture because in inverted microscope have objective at the bottom, unlike regular microscope. Light source is generally at the top. Animal cells are present at the bottom of the culture flask and growth occur from the bottom sides. So it is easier to observe through inverted microscope. Next question is, why we use refrigerated centrifuge for isolation of animal cell during animal cell culture. You know, animal cell culture is very delicate and sensitive to heat. During centrifugation, heat released by rotation of rotor and motor of centrifuge can damage animal cell culture. That is why cooling refrigerated system is attached in centrifuge for proper and normal isolation of animal cell for animal cell culture study. So this is very important question related to your CBSE exam. Next is, why it is preferred to or recombinant human erythropoietin instead of blood transfusion to a patient suffering from anemia. The main function of erythropoietin is just to make RBC. If the gene responsible for erythropoietin is defective, then no formation of RBC occur in the patient and patient will suffer from anemia. But if you inject the recombinant erythropoietin, that is can be produced through recombinant DNA technology and animal cell culture, if you inject in the patient, then there will be a proper formation of RBC occurs and there is no need of donor for blood transfusion. You can meet the requirement of blood, that is RBC, by blood transfusion. So no need of blood transfusion occur if you are injecting recombinant erythropoietins. So that's why there is no need of donor, no need of transfusion facilities are needed, and there is no chance of infection from the donor sites. That is why we prefer to inject recombinant erythropoietin instead of simple blood transfusion for making RBC. Next question is, why the need of myeloma cell with spleen cell during hybridoma technology? You know, myeloma cell is immortal and it can increase the lifespan of a spleen cell to produce more amount of monoclonal antibody for a longer period of time. That is why the need of myeloma cell with a spleen cell during hybridoma technology for maximum production of monoclonal antibody against a specific antigen, a specific viruses, a specific bacteria. Next question is, why OKT3 is required for a patient during kidney transplantation? OKT3 is the product of hybridoma technology. OKT3 is therapeutic monoclonal antibody that masks the function of CD3 proteins present at head of T cell of CMI system. So CD3 protein is responsible for recognizing the foreign tissue or organ rejections in human body. That is why OK3 is required for a patient during kidney transplantation for successfully. Why TPA is required? Why TPA is required for a patient having a stroke or brain hemorrhage? The main function of TPA is just to dissolve the blood clot of arterioles or artery. When there is a formation of blood clot inside the artery at the time of a stroke or brain hemorrhage or at the time of accident. So they, there will be proper circulation of blood will occur inside the brain or in the heart. Thank you.